ganglia and how it fails in Parkinson's disease. So let's get started. This is not anatomically correct, but it shows basically our general classical understanding of the basal ganglia function and motor function, and that's basically the direct is increases movement, and indirect is supposed to decrease or stop movement. But we know that both pathways lead to movement. The indirect pathway simply causes less movement or undesired movement. So first of all, before we look at this, I'm just going to draw something in here. The basic motor function of the basal ganglia, as we know it, is that your lower motor neurons from your prefrontal cortex, decision-making center, they first consult the basal ganglia. So it's prefrontal decision area, somewhere around here. They first consult the basal ganglia, and then the thalamus has spiny projection neurons that go all through the brain to the cortex. So, and then everything in red is a basal ganglia structures. So basically what's going on is your prefrontal motor cortex, decision making, goes to the basal ganglia to see to engage in voluntary movement and positional changes. And based on what the thalamus sends back, the lower motor neurons then go ahead and go down the spinal cord and initiate the voluntary movement. Now, the ventral pallidum is one thing that's not really involved in it. Kind of involved in motor, but it's more involved in addiction. So we're gonna have to clear up a few things. The mesolimbic pathway, limbic means memory, so the VTA, or the ventral tegmental area, it sends dopamine to the nucleus accumbens, and this is different. This is more what we think causes addiction, and the nucleus accumbens sends nuclei to the lateral thalamus, and that's so this is more known for reward processing versus we typically associate dopamine with addiction, but really it's motivation, focus, movement, memorization. So the substantia nigra contains a lot of dopamine projection fibers that go to the putamen and the caudate, which is called the dorsal striatum, because the nucleus accumbens, which is here, some people call it simply the anterior striatum, but it's really basal rostral to basal to all the other structures of the brain. So the substantia nigra, pars compacta, and pars reticulata is more GABA, but the dopamine, this dark area is here. That's where all the dopamine comes from. So basically, it sends its spiny projection neurons to the globus pallidus internal in order to create glutaminergic excitation, and you get more excitatory movement. Whereas in the indirect pathway, D2 receptors, they project to globus pallidus internal, and they still cause movement, but not as much. This is a gross section, so to the right is someone who had Parkinson's disease. You can see the substantia nigra is pale, and to the left is normal. You see the dopaminergic neurons, and it's dark because it contains melanin pigment as well. So we already talked about nucleus accumbens and dorsal striatum, right? The caudate nucleus is easy to remember because it's shaped like a C. And the thalamus, I don't know why it's blue here because it's sort of part of the basal nuclei, as it sends the spiny projection neurons back to the prefrontal cortex for it to make a decision. And so if, like, I call this is the goat of the direct and indirect pathways. So both pathways, remember, whether or not it's GABA inhibitory or glutamate excitatory, are going to increase movement, right? It's just that the D2 pathway is going to create less or promote undesirable movement like tremors, and I'll talk about this later when we get to Parkinson's disease. The only thing is the substantia nigra compacta sends dopamine. So let's look at this direct pathway here. So cortex is going to release glutamate, which is excitatory, right? And it's going to, what's going to happen is it's going to send it to the caudate, which is going to send so it's going to send to the caudate, which then goes through to the, because it's excitatory, it's going to go through the globus pallidus internal, and also it's going to go straight to the substantia nigra, which sends dopamine to the thalamus, which stimulates the inhibitory nuclei. So you get more glutaminergic input in back to the prefrontal cortex, and then the lower motor neurons direct more goal-motivated actions, and it's also involved in cognitive things as well, such as focus and motivation. Now, the indirect pathway, the motor cortex or the cerebral cortex, gray matter, still sends glutamate to the caudate here, right? But the indirect pathway has the substantia nigra send D2 receptors, which are the D2 it contains D2 receptors, but they're more inhibitory to the caudate than D1. So you see this glutaminergic excitatory input, and then this GABA inhibitory to the globus pallidus external, and then it goes on to the thalamus through the globus pallidus internal. And then since GABA inhibits the inhibitory, you get increased movement, just not as much, right? So it's the same thing, and it tells the subthalamic nuclei to go to the globus pallidus internal, to excite glutamate, release of glutamate, right? And the external inhibitory pathway goes to the subthalamic nucleus as well, you see here. And then this D2 inhibitory, and through, you can see through the arrows, it's confusing between direct and indirect, excitatory and non-excitatory, but regardless if it's GABA or dopamine direct, D1, it's going to disinhibit the thalamus, which is the inhibitor. So here, it's going to inhibit, D2 is it's going to inhibit, but the inhibitor, so it still creates movement, just indirect. And this is very important. So like this, back to the so simply put, motor cortex sends glutamate to the subthalamic nuclei, and then that causes to the globus pallidus internal, excites it with glutamate, and it sends GABA to the thalamus to disinhibit the inhibitor, and you get the motor cortex excited. And so, and then here, if it goes to caudate putamen, the glutamate, it also causes GABA to the GPI and GPE and the subthalamic nucleus, but the subthalamic nucleus sense nerve fibers with glutamatergic excitement, and if it's excited, the GP, globus pallidus internal, knows, okay, in order to excite it, I need to inhibit the thalamus, right? So it all may seem very complex, but it's actually quite simple. It's involved in voluntary action, and here's a Parkinsonian gait. I won't make you watch the whole thing, but this guy does a very great job. So in Parkinson's disease, once you lose all your dopamine projecting neurons, your prefrontal cortex is saying, move your right foot, move to walk, right? But there's no dopamine in the substantia pars compacta, which is the primary dopamine projection to the dorsal striatum. So that's why they take little steps, and sometimes they have freezing episodes, and sometimes they take that step because there's still enough dopamine for the motion. Now remember, this is different from dopamine that's we know is involved in addiction through the limbic system. That's why you can't just give someone with Parkinson's disease amphetamines because they cause release of dopamine, not in the nigrostriatal pathway, right? But the mesolimbic pathway, right? Reward processing for diverse functions. So you typically in a non-addict, reward processing includes things like memorization of knowledge and teaching and reaching goals and setting plans, but if you use amphetamines and the nucleus accumbens connects it with motor functions of like exciting activities. So if you go, for instance, if you do an exciting activity on amphetamines rather than study, you're more likely to become addicted because your limbic system will remember that, and the greater the exposure, the more you become addicted. Now, here's a study, interesting study that using you to prove that our classical model is wrong of the basal ganglia. So what they did was they labeled they labeled using GCAMP3 D1 cray. D1 cray means just knock in mice, so they knock in D1 in the direct pathway, and then in the indirect pathway, the A2 adenosine 2A cray mouse is just injected with this marker indirect pathway to knock out D1, and then they labeled it so they could so the fluoroscope could see the activity as the pulsed laser would send excitatory signals to the dorsal striatum. So you can see here it's sent to a D1 cray knock-in mouse, direct pathway. You can see that it's lit up, the fluorescence is lit up all the way to the substantia nigra, which makes sense, right? We have the globus pallidus internal activated, and the striatum is activated, and basically it sends its nuclei glutaminergic input to the thalamus to create motion. Now in the indirect pathway in the knockout mice, you can see it's a lot less fluorescence, but there's still fluorescence. There's still fluorescence, and the indirect spiny projection neurons, they express D2 and adenosine 2A, but for movement as well, just not as excitatory movement as glutamate and D1, right? So so you can see that there's appears to be less activity at the substantia nigra pars compacta. But so what they say that what this experiment did is they did a simultaneous pulsed laser excitation of direct and indirect labeled structures. And what they found was that the striatal neurons are co-activated during movement initiation and are inactive when the animal is not moving. So striatal neurons are inactive when the animal is not moving. These data call into question the predominant current model of basal ganglia function, which postulates there should be more activity in the direct pathway during movement than during immobility, and more activity in the indirect pathway during immobility than during movement. But what we know now is that there is more activity in the indirect pathway during movement. It's just that it's unwanted movement, like tremors and like the Parkinsonian gait, how they have the pill rolling tremor. It's so if you completely knock out the substantia nigra pars compacta, and you get no primary dopamine striata projections, what happens is the indirect pathway takes over, and that's why people with Parkinson's disease they'll sort of shake and move around, because they're not moving, they're still moving, but undesirable movements. And this part of the study just shows that the classical theory of the basal ganglia was that the direct and indirect are opposing effects on movement, right? But here, what they found was that whether or not a freely moving animal is excited with the laser during the inactive or active, or indirect or direct, you can see that it's still inactive. The D1 cray is inactive in the direct, and the same thing you can see in the indirect. It's a little less activity, as we would suspect, but it still holds that the voluntary movement was more active even in the indirect pathway, right here, and more so in the direct pathway, because this is D1 and this is A2A and D2. So, and then what's even cooler here is that what they found was that before movement is even initiated, what's super cool is that at the same time that you're thinking about initiating a movement, your basal ganglia already sends the signal to initiate the movement not before you think about it, but like 500 milliseconds before, right after you think about it, the basal ganglia already says how much of excitatory movement it's gonna cause. So it shows activation immediately before and after, so which makes sense, but yeah, so basically your prefrontal lower motor neurons, before they go on and do their thing, they consult with the basal ganglia, because the basal ganglia needs to, depending on its function, as long as the function is normal, the direct pathway will be the primary pathway, and the indirect pathway will also, in conjunction, also work, but it will not be as stimulatory as the direct pathway. That's it in a nutshell. It's once you can wrap your head around this, it's actually not that difficult to understand. And with that, thank you for watching. Leave any comments. This is an extremely complex topic, but very interesting to discuss.